think we can start to get in. It's wonderful to be here this morning and to introduce somebody who is absolutely exceptional and who is newly recruited to our department in neurosurgery and also to Smilo and the Cancer Center. Dr. Roel Verhaak is professor in the Department of Neurosurgery at the Yale School of Medicine. Following graduation with a PhD in medicine from the Erasmus University Medical Center in Rotterdam, the Netherlands, Roel joined the Broad Institute, Dana-Farber Cancer Institute, as a postdoctoral associate supported by a fellowship from the Dutch Cancer Society. During the time at the Broad, he was part of the team analyzing data from the TCGA. He led the identification and characterization of gene expression subtypes in glioblastoma, work that resulted in a seminal Cancer Cell 2010 publication. Roel moved to MD Anderson Cancer Center in 2010 to start his own laboratory. Since then, the Verhaak lab has studied tumor evolution and mechanisms of therapy resistance in low and high-grade gliomas. The group was foundational in establishing the Glioma Longitudinal Analysis Consortium, which has established a resource of molecular profiles over time on a large cohort of patients with glioma. They identified and described genetic scars and cellular phenotypes associated with glioma progression and disease recurrence. Extrachromosomal DNA amplifications were discovered as critical drivers and are now a major part of the team's research. After being affiliated with the Jackson Laboratory for Genomic Medicine in 2016, I can tell you our department leadership fought very hard to recruit him here to Yale. And he joined us in the Department of Neurosurgery in 2023. Roel is a recipient of the AAAS Watchell Award, the Agilent Early Career Professor Award, and the Peter Steck Memorial Award. He's also co-founder of Boundless Bio. I can tell you in the short time that I've had the privilege of working with him, he's truly exceptional. And I'm really excited for this talk and for all of the work that we have to come. So without further ado, thank you so much, Dr. Verhaak.
Thanks, Jen, for that very, very kind introduction. And so I joined the Department of Neurosurgery in April of last year after some discussions with Dr. Grinnell and others. And to be honest, it wasn't that hard. I was pretty convinced very quickly that this was going to be a great place to continue our research. As we were thinking about becoming more translational, we felt that being in a clinical environment would greatly benefit our work. And what greater clinical environment than Yale School of Medicine and Department of Neurosurgery. So I'm a co-founder of a biotech. I won't be discussing that work today. I am also a consultant for neurotrials. So gliomas are the most common malignant tumor type in adult patients and the most devastating ones. They're characterized by an infiltrative growth into the environment, into the parenchyma. And this makes these tumors exceptionally hard to treat because you can't go in with a knife and cut out the entire structure for obvious reasons. Nowadays, we recognize, traditionally, we would classify gliomas in adult patients by histopathology. Fortunately, we've gotten away from that as molecular markers do a much more precise job in doing such classification. But nowadays, we recognize gliomas based on two critical molecular markers. First, we identified the presence or absence of a mutation in IDH1 or IDH2, isocitrate dehydrogenase. And for those cases that carry an IDH1 mutation or an IDH2 mutation, we further separate them by the presence of or absence of 1p 19q co-deletion, so chromosome arm loss of 1p and 19q. Predominantly, the cases that have this co-deletion, we call them codels, are in majority histopathologically oligos. The non-codels, so those cases that are IDH mutated but don't have that co-deletion, are in majority astrocytomas, and the IDH wild-type cases are mostly the glioblastomas. And the patient's survival patterns are according, meaning that those cases that have no IDH mutation do particularly poorly clinically. That's not to say that any of these tumor types are better to have, quote unquote, as far as you can ever better have a tumor. Patients that carry the IDH mutant non-codel tumors are typically diagnosed between 35 and 44 years of age, so very young in life. The codel patient typically is around 45 years of age. So again, relatively early in life. So those patients might have much better outcomes, but they'll mostly, the majority will still succumb to disease even prior to the median age of diagnosis for IDH wild type tumors, which is around 60. So these are all bad tumors. Here's the motivation for classifying gliomas by these two molecular markers. In part, it's of course it's clinically, it's the survival outcomes, as I showed you on the previous slide. But here in this paper from the TCGA from 2015, we demonstrated that not only behave these tumors differently and they respond different to treatments, but they're really biologically different entities as reflected in the sets of molecular alterations that are commonly detected. For example, in the CODEL group, we find that they are nearly universally carrying mutations in the TERT promoter, as well as relatively spurious mutations in genes like NOTCH1 and CIC. The non-CODELs, even though they are IDH mutated, rarely contain TERT promoter mutations, but are universally mutated in p53 and 75% carry alterations in ATRX. So very similar tumor types, but molecularly quite different. And then finally, IDH wild type tumors, again, 80% are TERT promoter mutated, and they are then a majority containing mutations in genes like EGFR, CDKN2A, PTEN, and so on and so forth. So biologically, not just responding differently to treatment, not just different in terms of when they present in life, but biologically also quite different. Now, after we were able to refine the classification of gliomas in adult patients, a major challenge continues to be how these tumors respond to treatment and the lack of new treatments coming into the clinic. Now, tumors initiate from a cell of origin, and over time, as these cells respond to challenges in the tumor microenvironment, for example, presence of oxygen, lack of nutrients, and so forth, you'll find that intratumoral heterogeneity starts to develop. And this is a consequence of these evolutionary processes and clonal selection, where some cells are better able to deal with these limitations than others. Therefore, they show clonal outgrowth. So at the time of diagnosis, we're dealing with an heterogeneous tumor with different sets of tumor cells marked by specific mutations and other kinds of gene alterations. Now, critically, this process doesn't end at diagnosis, of course. We impose treatments onto these tumors, surgery, initially debulking surgery, combined with radiation and chemotherapy, which for gliomas in majority is temozolomide. And of course, these treatments continue to impose these bottlenecks onto the tumor. And those cells best able to deal with radiation, best able to deal with chemotherapy, are the ones that are going to fuel the outgrowth and the tumor recurrence. So we felt that this would be an important process to study, so that we could try and make these treatments more effective and potentially identify targets for new treatment development. So with that in mind, we started the Glioma Longitudinal Analysis, or GLASS Consortium, in 2015. The GLASS Consortium set out to, it developed on the tail ends of the TCGA, and it set out to develop a comprehensive molecular reference data set from pairs of tumors obtained at diagnosis and then after treatment, so the first tumor recurrence. But in reality, we have been collecting tumors along the whole trajectory. So we have cases now for GLASS where we have 6 recurrences consecutively, and we've molecularly characterized them. In other words, we've sequenced them. And then critically for GLASS, we really try to curate and obtain clinical annotation for all cases in the cohort, because the value of a resource like this is significantly amplified if we know which tumors got treated in between time points. Now, why did we need to do a consortium for this? It's because of things like patient mobility and the way tumor banks work. If you go to your average tumor bank, surely you'll find some tumors for which there's multiple time point specimens. But even for MD Anderson, where we used to be, one of the largest centers in the country, we were limited to a few dozen cases where we would have these multi-time point specimens. And then as you're dealing with attrition due to tissue quality, at the end of the day, you really need an international collaboration to get to the large enough numbers to do any kind of robust analysis. So we started the consortium also still being enthusiastic of how well the TCGA collaboration went for us. And now have developed a consortium that involves over 140 people spread across the globe, essentially, in 14 different countries. This is an older picture, actually. If you would take a picture now, it would fill the room. So an important purpose of GLASS is not just to create this data set, but also to share it broadly, just like the TCGA, so that not just we can do interesting analyses with them, but of course that the whole community can do so. So in 2022, we released our latest public version of the GLASS data resource, which is a cohort of over 300 patients, for whom we have collected multi-time point DNA sequencing and/or RNA sequencing. Now, these 300 patients, and I can tell you in the meantime, now we're 2024, we've nearly doubled the cohort size and we'll be releasing that soon. So we continue to actively expand this cohort. Right now, in our cohort, if you go to the URL shown here, you can find variants in clinical annotation for over 300 cases, of which a majority are from IDH wild-type tumors, followed by the non-codels, finally the codels. As you can see, we have a relative underrepresentation of codels here, and that's likely due to the longer time to recurrence for codel tumors compared to IDH wild-type tumors. The shorter the time to recurrence, the higher the likelihood that two tumor specimens will end up in the same tumor bank. In addition to that, re-resection is not standard for any of these, so that's another factor that comes into play here. You can maybe appreciate that the median age of diagnosis in these three groups is relatively young, and that is because in order to end up, for data to end up in our resource, the patient has to have had 2 surgical procedures in order to obtain specimens, meaning that the patient has had to be in relatively good shape to be able to undergo those procedures. So we see a bit of a bias in median age at diagnosis as well as in survival patterns shown here. That is what it is. We can't really address that. We try to address it by expanding the resource as large as we can so that we capture as many patient groups as we can. Finally, I'm going to point out that our annotation, in my opinion, is really great. So we know for all patients, whether they or nearly all patients, whether they have received temozolomide, yes or no, and whether they have received radiation therapy, yes or no. And we've got many more clinical variables. I just chose to highlight these on this particular slide. So in my opinion, it's really becoming a phenomenal resource. Now, what can you do with a resource like this? I think you can do many things, but we initially focused on 2 important questions. First, what is the impact of temozolomide on tumor evolution and on these gliomas? And second, what is the impact of radiation? So treatment with temozolomide is a dealkylating agent. The repair process of the dealkylation can show up as mutations and nucleotide changes. Nucleotide changes can conveniently be detected using sequencing, and that means that in a subset of tumors that are treated with temozolomide, a hypermutation phenotype will develop. So these are tumors where cells have been able to overcome the damaging effect of temozolomide, and they do so by repairing the damage caused by temozolomide, and that damage is then showing up as hypermutation, very high mutational burdens. Across our cohort, and this is a slightly older version of our data set, but across our cohort, we then see that when we compare mutational burden of the initial tumor and the post-TMZ treated recurrent tumor, so this is only TMZ treated cases, we see very high mutational burdens in these recurrences, and this is a log scale, so we chose a cutoff of 10 mutations per megabase here. So across the 3 subtypes of glioma, we see that a subset recurs as hypermutated. The relative frequencies differ by subtypes. IDH wild-type tumors, 15 to 16%. For the IDH mutant tumors, we see that the rate of hypermutation development is much higher. We think this is due to the fact that IDH mutated tumors take longer to recur. Therefore, there's more of an opportunity for hypermutation to develop. hypermutation has been associated with relatively poor outcomes. What we found when we evaluated the presence of hypermutation in TMZ-treated tumors and then compared the time to progression, that it's actually similar between non-hypermutated, so tumors that did not become hypermutated, versus those that did become hypermutated. So time to progression doesn't really depend on the development of hypermutation. But once a tumor has become hypermutated, so after that recurrence, the hypermutators do worse than the non-hypermutators. That's not to say that we shouldn't be treating these patients with temozolomide. Clinical trials such as the CATNON study from the EORTC have clearly demonstrated that temozolomide has significant benefits across the patient population. So even though sometimes people will argue, well, temozolomide causes hypermutation, hypermutation is bad, as a patient group, temozolomide is clearly beneficial. Emre and Kevin in our lab then chose to study similar questions, but then for response to radiation therapy, which causes a different type of DNA damage. It causes single strand breaks as well as double strand breaks. And long story short, they discovered that when comparing cases not treated with radiation to those that are treated with radiation, that treated cases develop a relatively high number of small 2 to 20 base pair deletions scattered across their genome. So it's a bit of a similar phenomenon to hypermutation, but instead of single nucleotide changes, we found that radiation drives small deletions. And in the treated cases, we see significantly more small deletions arise compared to the untreated cases. Now, radiation and temozolomide are often used in combination. It's standard of care for IDH wild-type tumors. So are we observing this increase in small deletions because some of these tumors are developing hypermutation? The answer is yes and no, meaning that when we split up our cohort into those cases that are hypermutated as well as radiation treated, we find that hypermutation independent of radiation, actually, so these are tumors that are hypermutated and have not been treated with radiation, that they also show an increase in the number of small deletions. But importantly, those that are not hypermutated and have been radiated also show that small deletion increase. So hypermutation and radiation are independent factors driving the increase in small deletions. And in that sense, the small deletion increase, burden increase, is comparable to hypermutation for temozolomide. And in our paper, we actually found that this is true across cancers, not just gliomas. Emre and Kevin then also evaluated aneuploidies, in other words, broad losses and gains. So while small deletions will arise from double-strand breaks that are subsequently repaired, aneuploidies typically are a result of cell cycle of errors during the cell cycle. For example, missegregation. We compared broad gains and broad losses. We did that between irradiated cases and unirradiated cases and found no difference in gains. But we found a significantly higher number of whole chromosome arm losses in irradiated versus non-irradiated tumors, similar to the small deletion increase. Now, homozygous deletion of CDKN2A, which is of course a cell cycle regulator, has previously been associated with tumor progression, especially in IDH mutant tumors. We compared not just homozygous deletion, but also hemizygous deletion of CDKN2A, first in untreated initial tumors in GLASS. This is codels and non-codels, and this is focusing on the IDH mutant tumors. Codels versus non-codels first, and we find that non-codels have a higher rate of CDKN2A homozygous as well as hemizygous loss. But this is particularly pronounced in recurrences. So at recurrence, non-codel IDH mutant tumors show a significant increase in the number of CDKN2A homozygous as well as hemizygous deletions. And this is then particularly true amongst irradiated tumors, suggesting that there's a relationship between irradiation and CDKN2A loss. And again, the presence or when a CDKN2A loss, either hemizygous or homozygous, has been acquired, we see that that associates with worse outcomes to treatment. We then evaluated the association between these broad aneuploidies and CDKN2A loss. Here we grouped a bunch of cases. Actually, this is not GLASS data, this is pan-cancer data. We grouped those cases by non-irradiated, palliatively irradiated, so lower doses, and curatively irradiated tumors, and find that the increase in the number of chromosome losses is actually only found in tumors that have homozygous deletion of CDKN2A. So while we previously showed that irradiation appears to drive broad chromosome losses, what we're actually seeing is that irradiation associates with CDKN2A loss, and it's really the CDKN2A loss that then associates with aneuploidy, because we're seeing a significant increase in the number of chromosome losses in unirradiated cases when homozygous loss of CDKN2A is present. So irradiation itself does not appear to drive aneuploidy, it appears to drive CDKN2A loss, which then drives the aneuploidy. And as with the hypermutation example, we're finding no significant difference in surgical interval in irradiated cases associating with the number of acquired small deletions. But post-recurrence, those tumors that acquire the highest number of new small deletions are the ones that stop responding to further therapy, that have very poor outcomes. So acquired small deletions are a marker for further tumor response, if you will. So this is the model that seems to arise from our data, which is perhaps not super surprising. These tumors, as I started with, originate from a cell of origin that starts to expand more quickly than the cells around it. And upon bottlenecks in the tumor microenvironment, subclones will further arise that then at the time of diagnosis can be detected through sequencing. Then we impose this therapeutic barrier, first surgery and then chemo and radio. And those cancer cells that are able to repair the DNA damage, so the ones that have the hypermutation phenotype or the ones that have acquired large numbers of small deletions, those are the ones that are able to repair the DNA damage and can subsequently be detected, have expanded sufficiently, have become large enough subclones that we can detect them through sequencing. So then it's not surprising that at recurrence, these tumors that have these genomic scars, these signatures, are the ones that have poor outcomes and stop responding to treatment, because you're looking at cells that have already shown that they don't care about further DNA damage, or they don't care about chemo or radiotherapy. Now, when you summarize these numbers across our GLASS cohort, we see that amongst IDH wild-type tumors that have been treated with temozolomide and/or radiation, that 15% develops the hypermutation phenotype. And of those that do not develop the hypermutation phenotype, another 16% acquires large numbers of small deletion, which leaves a relatively large group in which no genomic scars can be detected at recurrence, and they may have intrinsic mechanisms to deal with the toxic effects of therapy. When we look at IDH mutant tumors, where the picture is more diverse because not all patients will receive the same therapies, we find that amongst those that have been treated with temozolomide, 42% acquires hypermutation, 35% of non-hypermutators acquires the small deletion phenotype, and again, a subset shows neither. Now, as Jen mentioned in the intro, in previous work, we have looked at gene expression patterns, and this is focusing on GBM, so IDH wild-type tumors. And we found that when we evaluate gene expression patterns, we can identify 3 gene expression subtypes of glioblastoma, IDH wild-type glioblastoma, which we labeled mesenchymal, perneural, and classical. When we evaluate subtype classification in GLASS, we see that the relative distribution of these 3 subtypes is what we typically would expect. A number of cases are classical, mesenchymal, or perneural. At recurrence, we do appear to see a minor shift towards mesenchymal tumors. So we see a higher number of mesenchymal tumors at recurrence. So tumor progression appears to, in some tumors, coincide with mesenchymal transformation. But perhaps more importantly, we find that these subtypes identifications, classifications, are quite flexible because nearly half of our cases actually change subtypes between the initial time point and the recurrent time point. Now, like many tumor types, glioblastoma has been extensively studied using single-cell sequencing and single-nucleus sequencing. Our collaborator Mario Suva has led the field in this respect and published a very influential paper in 2019, the Neftel et al. Study, in which they found 4 predominant cell states of glioblastoma, which they labeled oligoprogenitor cell-like, neuroprogenitor cell-like, astrocyte-like, and mesenchymal-like. And you may notice that these terms are actually reminiscent of the subtypes that we had previously identified. And that's maybe not surprising. If a tumor contains a majority mesenchymal-like cells, the subtype signature will be mesenchymal. We've shown this using combined bulk RNA-seq and single-cell RNA-seq data sets. Oh, and another important thing to remark here is that all GBMs, each of the cell states can be detected in all GBMs. It's just a shift in numbers, which perhaps explains the plasticity of expression subtypes over time. Now, my lab, Kevin Johnson and Kevin Anderson have worked together to do single-cell sequencing of gliomas as well, with the purpose of identifying pan-glioma cell states. Mario's Neftel et al. Cell states are focused on IDH wild-type tumors. Our effort initially focused on identifying cell states that could be identified across different types of glioma. And we found those, we labeled them stem-like cells, proliferating stem-like cell, and differentiated stem-like cell. And of course, with single-cell sequencing, you can also identify non-malignant cell states, such as oligodendrocytes, parasites, myeloid cells, cells that are typically residing in the microenvironment of these gliomas. Now, you can infer signatures, gene signatures from these single-cell states. And you can then use computational methods to project those signatures on bulk RNA-seq datasets, such as the ones we have in GLASS. So you can use single-cell signatures to deconvolute bulk expression profiles. Sir Fred Warren, former postdoc in the lab, now a faculty member at the Jackson Laboratory, took this approach, used the single-cell inferred gene signatures from the Kevins, and projected those onto the GLASS RNA-seq datasets. This is showing on the left the summary of IDH wild-type tumors, on the right the IDH mutant tumors, primary and recurrences. So when we aggregate all the data, first starting with IDH wild-types, when we aggregate the presence of these single-cell signatures across the cohort and compare initial to recurrent tumors, we do not find major shifts in our pan-glioma cell state presence. Actually, the major difference we found when comparing initial tumors to recurrent tumors is the relative fraction of oligodendrocytes in the tumor microenvironment. And maybe this is not too surprising. If you look at the invasive margins of these tumors, this is at the time of recurrence, this is where most of the tumor cells will have come from, because this is the area of the tumor that's difficult to resect. So perhaps at recurrence, you can imagine that at recurrence, more of that margin is cut out, and therefore we might be able to see more cells from that microenvironment, in this case, particularly oligodendrocytes. What did seem more surprising to us is then, again, when Fred used computational methods to not just enumerate the types of cells in primary to recurrent tumors, but also looked at the actual gene expression profile of those cells, that we found that a significant increase in neuronal signaling pathways amongst the malignant cell population. Of course, you'll find neuronal signaling in oligodendrocytes, but what we were finding is that also the malignant cells activate neuronal signaling pathways at recurrence. So we're seeing an increase in oligodendrocytes in the microenvironment, but that appears to be converging with increased levels of neuronal signaling by the tumor cells. And when we used a public data set consisting of multi-biopsy single-cell RNA sequencing from glioblastoma patients, we could again confirm that the malignant single cells expressed higher levels of neuronal pathways when the biopsies were obtained from the margins of the tumor relative to the core of the tumor, confirming what Fred had found in our bulk analysis from GLASS. I'm actually going to skip this one. We decided to then take this one step further in a large collaboration that involved Mario Suva, Itai Tirosh, Antonio Iavarone, and Anna Lessarella, as well as many postdoc and junior leads in these respective labs, collaborating with MD Anderson, Duke, and a number of other institutions. We generated longitudinal single nucleus RNA-seq data for a large number of IDH wild-type glioblastomas. Again, in the context of annotation for different types of therapy. And we also were able to generate exome or whole genome sequencing on the majority of our cohort. So previously, Mario and colleagues identified these 4 cell states that I mentioned earlier: NPC, OPC, AC, and MES-like cells. When we analyzed over 500,000 cells from this cohort, and again to derive cell states as well as transcriptional meta programs, we find these same 4 cell states and the gene expression signatures that come from them again. Here is OPC, AC, mesenchymal, and NPC. But of course, we found many more because of the much larger cohort, as well as because in Mario's initial study, he included only untreated tumors. And now we're looking at both primary and recurrences. So our large data set enabled us to find a number of new glioblastoma-related cell programs. As we had also observed in our GLASS analysis, the relative number of malignant cells decreased at recurrence. So recurrent GBMs become less pure or more incorporate more tumor microenvironment cells. So we see a decrease in the number of proportion of malignant cells, and an increase, and we confirmed the increase in the number of oligodendrocytes. But because of the greater resolution of the single cell, of the new single nucleus data, we also see a significant increase in the number of astrocytes and the number of neuronal cells. Converging with the result from GLASS that most tumors or many tumors change tumor subtype at recurrence, we find large shifts in cell states between primary and recurrent tumors. And the one that maybe is interesting is hypoxia, and I'll get back to that later. So a subset of this smaller cohort compared to GLASS acquired this small deletion phenotype that I mentioned earlier. In fact, 10 of 46 tumors where we had converging DNA sequencing and single nucleus sequencing data acquired this small deletion phenotype as shown here. And what we're seeing is that when a small deletion phenotype has been acquired, tumors will increase. We find that more tumor cells show signs of hypoxia, are responding to hypoxia. So radiation either drives or shows its most significant effects in cells in regions of hypoxia. When we then went back to our GLASS data sets, we could confirm that indeed tumors that acquire lots of small deletions are also showing an increase in hypoxic signaling, hypoxia cell state signaling, compared to tumors that do not acquire small deletion. This is potentially relevant because hypoxia is a phenomenon you can detect through imaging. And of course, these results build upon a large amount of literature demonstrating the convergence of radiation response with hypoxia. We then also performed a comparable analysis, but looking at IDH mutant tumors. So we generated single nucleus RNA-seq data on IDH mutants, a cohort of 35 cases. And this is work led by Kevin Johnson, who is a research scientist in our lab, again with the same collaborator team. Mario and Itai's labs have previously found that in IDH mutant tumors, they found less consistent cell state and gene expression programs, but they found that that most tumors could be projected along an axis of stem-like to stem-like signal from stem-like states to a more differentiated state. Because IDH mutant tumors in general are either astrocytoma or oligodendroglioma, they showed that astrocyte IDH mutant gliomas differentiate from a stem-like cell to a astrocyte-like cell phenotype, whereas oligodendrocytes go from a stem-like state to a more oligodendrocyte-like state, as potentially expected. So the non-codels go to the left and the codels go to the right. In our paper from 2022, with Fred as first author, we had noticed that amongst IDH mutants, those that show signs of treatment response either through hypermutation or through acquired CDKN2A loss, we saw an increase in the proportion of proliferating stem-like cells, which would be in the trunk of the axis shown on the left. So we took those results we took those into consideration as we started to analyze these data. So first, Kevin generated these UMaps that you can see in many papers. We had generated data from large numbers of nuclei, I would say quite unprecedented, to show that you can infer your typical sets of cell state programs as shown here. So we have now generated a very large number of IDH mutant single nucleus data. And through that, we can create a definition of cell states and associated meta programs of IDH mutant tumors. And the meta programs we arrived at actually are quite reminiscent of those that are shown in IDH wild type tumors. Interestingly, when we looked at when we projected the 35 cases that we had analyzed, that we had sequenced, and projected them on that same inferred y-axis as Mario and Itai had previously used in their analysis, we see that tumors tend to shift. So the circles here are the initial tumors and the triangles are the recurrent tumors. We see that nearly all tumors shift into an upward direction. So the relative amount of the relative amount of stemness in these tumors almost universally increases upon recurrence. So tumor seems to de-differentiate as a part of their tumor progression. This is also shown here. These are different, some of the different meta programs that we had arrived at, looking at different grades amongst codels and non-codels. And we see that a differentiation cell state, such as the AC-like state, decreases with grade, and that's true for both codels and non-codels. Whereas undifferentiated and the number of cyclin cells increases with grade. And then when we actually pair up the tumors, so not split them by grade, but actually look at paired samples, again, we confirm that the amount of undifferentiated cells increases, the amount of cycling cells increases. Cells that show signs of stress increases in proportion, and finally, mesenchymal-like cells increase in proportion. Now, what is, in my view, most interesting about these observations is when we now separate our, relatively modest cohort, but still, into cases that have signs of therapy-induced genetic alterations, that those are the ones that those are the ones that are driving these significant changes. So tumors that do not reflect therapy-induced genetic alterations, such as hypermutation, CDKN2A loss, or loss of new aneuploidies, we find no significant difference in cell states. It's only those tumors that show a treatment-induced alteration that are the ones that also show changes in their gene expression programs. So to then re-annotate the flowcharts I showed earlier, we're seeing that subsets of IDH mutant as well as IDH wild-type tumors acquire genetic alterations in response to treatment. And we're now finding, using our bulk and single nucleus gene expression data sets, that this coincides with increased cell cycle activity and proliferation and de-differentiation programs in IDH mutant gliomas, but with neuronal and mesenchymal signaling activity in IDH wild-type tumors. So it leads me to summarize at the end here. IDH wild-type gliomas, so far, seem to undergo tumor cell extrinsic changes, which sets them apart from IDH mutant gliomas, which appear to in majority undergo tumor cell intrinsic transitions, which I think is a peculiar but interesting difference. As we think about developing new therapies for these patients, this is something to take into consideration. And finally, amongst the IDH mutant gliomas, the changes we are observing are mostly observed when in those tumors that have been treated. And we find convergence between newly acquired genetic alterations with cell state transitions. So that leads to the question, are these tumors changing because of the treatment, or are the oncologists treating the tumors that are more likely to change, or both? That's something for a next analysis. With that, I come to the end. I'd like to thank all the people in the lab that work very hard for these results, and of course our funders. Thank you very much.
Thank you. Jen had to run to the OR, so I will handle the questions. Do we have any questions from the room or online? You mentioned immunotherapy. So are there protocols now that are using some of these markers to determine who should get immunotherapy and which ones in this disease?
So regrettably, all the results so far have shown that checkpoint inhibition does little for these patients. And that's likely because of the very immunosuppressive microenvironment in these tumors. There's very few active T cells. So you can treat them with checkpoint inhibition, but without T cells, that's going to not really result in any benefit. So moving forward, the way to get immunotherapies to work in these patients would be to figure out how can we get T cells into the tumor, and only then immunotherapy is likely to have a chance.
Got it. Okay, any questions in the room? Okay, I will walk the microphone around. I'll go to the front row here first.
Hello. Oh, thanks. A beautiful talk. And I think, just it's something that we are all hoping to be able to replicate in different tumor types. What a great example of a treasure trove of information. My question is about epigenetic regulation, and I saw one slide with EZH2. Your thoughts, or if you've looked at sort of wrapping of chromatin, epigenetic regulation, specifically after radiation, if that's changed, and if we can explore that with some of our, for example, EZH2 or other regulators there, inhibitors there.
That's a great question. So just from a data perspective, we have been able to collect DNA methylation profiles. Other members of the GLASS Consortium have looked at those. What we see in the IDH wild-type tumors, we don't see many changes from a DNA methylation perspective. These tumors have lots of things going on, but it doesn't really seem to change directly their DNA methylation profile. In the IDH mutants, we see that a subset of tumors goes from a relatively high amount of genome-wide DNA methylation to a decreased amount. So, and those are the ones that are also the ones that acquire genetic alterations that change cell state programs. Those also seem to demethylate or show demethylation genome-wide. Now, whether that has implications for treatment with EZH2 inhibitors would be a bit of a stretch. I know those are being considered for the H3 wild-type pediatric GBMs, for example. But right now, I don't have information on whether that would work for adults.
Great. We could go to Dr. Kropp, and then Dr. Contessa in the chat has a question, so we'll get him queued up to ask it verbally. Ian.
Very nice talk. And this question actually is a little bit similar to, I think, what Joe's getting at. So you've shown that in the subset of the temozolomide-treated patients develop this hypermutated phenotype, and that leads to poor outcomes in those patients. it would seem that if you could potentially, if you could identify those patients up front who are going to go down that path with treatment with temozolomide, that you may decide it may be in their overall better outcome to avoid using temozolomide in those patients. So if you looked at baseline molecular genomic characteristics of the patients who go on to develop hypermutator phenotype, to be able to, if you could predict those up front?
Yeah, so for IDH wild type, of course, we have a great marker. It's MGMT methylation, right? So for that, I would say that's already largely addressed. For IDH mutants, we have not looked at this very much yet. There's been another publication from a group in China that has established a large number of serial cases. They have suggested that low-level changes in chromosome 8 would be predictive of risk of developing hypermutation, and they link that functionally to MYC. I think that data is interesting. I think it could use some further validation. Now, as we are expanding and working on our GLASS effort, a major change relative to our latest release and one we are working on right now is that we've accumulated a large amount of whole genome sequencing data. And I'm excited about that because with whole genome sequencing data, you can do things with mutational signatures. And mutational signatures would reflect, for example, potentially DNA damage repair processes that are ongoing in these tumors. So I'm hopeful that we can identify tumors that have DNA damage repair processes going on, and that would then be predictive of response to temozolomide. That's all speculation. So hopefully in a year from now or so, we will have more definitive answers.
Thanks.
Great. Dr. Contessa, I'm told we don't have access to allow him to talk.
Can you hear me?
Ah, a
Okay.
miracle. Yeah, okay, go ahead, Dr. Contessa.
Oh, great, Roel. That was fantastic. Fantastic talk. Very exciting. So yeah, I just wanted to drill down a little bit on the radiation-induced mutations, because there is this question, right? Is it that that after radiation, it's a selective pressure, and you're winding up, finding those mutations that have gone on and been propagated in different clones? I think that the main question is, so if you're sequencing from a tumor and considering the stochastic nature of radiation, do you think you're going to be able to find those recurrent, small deletions? And isn't that probably more consistent with you have a resistant clone, which might be, have a DNA repair defect, which enables radiation resistance, and so then you wind up having that radiation resistant clone moving on, and I think that's similar to what you would see with CDKN2A, but I won't be too long. And I guess my main question is, so you have these two different possibilities. Could you use a single cell analysis to try to differentiate between, right? Is it the radiation that's the cause, or just the that it's the selective pressure?
Yeah, thanks very much, and great question. So if we take hypermutation following temozolomide as an example, because of the specific mutational signatures of mutations acquired after temozolomide, we're pretty sure that temozolomide is actually causing these changes. And I think there's a lot of similarities between the small deletions acquired by after irradiation to the temozolomide example. One reason for saying that is that we have taken cell line models and irradiated them and then passaged them for 25 times or so, or have made sure they went through a full cell cycle 25 times over a period of, let's say, 3 or so months. One of our MD PhD students has done that in the lab. And she showed that after about 3 months, you see a significant increase in the number of small deletions in tumors in cell lines with irradiation versus those that have not been irradiated. And she actually spoke with your student after her exciting talk just 2 weeks ago. So that, to me, suggests a pretty strong causal link. Also, the types of small deletions, we've now made some progress in analyzing them. They carry a specific signature, or they are associated with a specific signature, which again, to me, suggests that there's a direct causal link rather than radiation causing clonal outgrowth of a particular clone. Yeah, I think that's what I wanted.
Yeah, thanks. We should connect, because I have some more comments and discussion.
Great, I would love to.
Okay, I just unmuted Dr. Robinson. Do you want to ask your question?
Yeah, phenomenal talk. I was going to ask, I think you already answered this about the MGMT methylated, if there's a difference in patterns of resistance, but The other question I was going to ask is, have there been efforts to kind of pursue synthetic lethal screens of some of these identified pathways, so CD&K, things like that?
It's a great suggestion. My speculation is that probably somebody has done that. We're not doing those in the lab right now. I guess challenges of course exist, even though challenges exist of course with getting any kind of molecules into the brain. If you have a target, many of our clinical trial failures that we've seen so far are actually related to blood-brain barrier and things like that. So I think your idea, of course, is very good. It will take a little bit longer before we can actually see drugs and treatments materialize from that.
And one follow-up question. One thing that's always really been perplexing to me is that with EGFR3 variants, is that if you put those in a petri dish, those get selected out. So they're actually disadvantageous in a petri dish, but obviously we see them in real human tumors. Do you have any kind of sense or any insights as to why those are advantageous in the real human environment, but they're not in a petri dish?
So it's hard to answer that directly. Maybe it has to do with the types of ligands that exist in the microenvironment versus those that exist in a petri dish. The spin I would give on this is that we find that all V3 variants exist on extrachromosomal DNAs. So EGFR is amplified when the V3 is present, and these amplifications typically reside on extrachromosomal DNAs, and there's a lot of that does a lot of things to these cells, including potentially putting a higher burden on the cells to produce all the DNA needed for the high numbers of copies that typically exist when there's extrachromosomal DNA. So that could be one reason. And in general, I think ecDNA is very potent in many ways, and that probably has to do with why these are selected out, more so than the V3 itself.